And let me now introduce our next speaker, Professor Randy Schekman.
It's me. Okay.
So our next speaker is Professor Randy Schekman. He is a professor in the Department of Molecular and Cell Biology here at Berkeley and an investigator of the Howard Hughes Medical Institute, or HHMI. He studied enzymology of DNA replication as a grad student with Arthur Kornberg at Stanford University, and his current interest in cellular membranes developed during a postdoctoral period with S.J. Singer at UC San Diego. Schekman's laboratory investigates the mechanism of membrane protein traffic in the secretory pathway in eukaryotic cells. In recent years, his lab has turned to aspects of vesicular traffic in human cells, most recently on the biogenesis and sorting of small RNAs into extracellular vesicles. Among his awards are the Gairdner International Award, the Albert Lasker Award in Basic Medical Research, and the Nobel Prize in Physiology or Medicine, which he shared with James Rothman and Thomas Südhof. From 2006 to 2011, he served as editor-in-chief of the Proceedings of the National Academy of Sciences. In 2011, he founded and until 2019 served as the editor-in-chief of the open access journal eLife, sponsored by the HHMI, Wellcome Trust, and Max Planck Society. Beginning in 2018, Schekman assumed a leadership role in an effort supported by the Sergey Brin Family Foundation to identify and support basic research on the mechanisms of Parkinson's disease initiation and progression. So without further ado, Professor Schekman.
Thank you very much. Well, I'm pleased to tell you a little bit about my background and just a snapshot of my research. And I'll spend more time talking about a newer initiative that I've become involved in on Parkinson's disease that I've approached from both a professional and personal angle. So a little bit about myself. I grew up in Southern California. I went to high school in Anaheim, near Disneyland. I went all the way into Westwood to go to UCLA during the greatest college basketball era of all time, when UCLA won a number of national championships in a row. I did a brief I did my graduate work at Stanford, for which I've been caused to apologize for the years that I've been here, but it was nonetheless a good experience. But I've been here on the faculty since 1976. I've never left. Next year will be my 50th year on the faculty. So when I was a kid, I grew up with, for some reason, a fascination with microorganisms. I had a toy microscope that I got when I was maybe 11 years old. And I remember collecting a jar of pond scum from a local river and put a drop of this on a slide and was amazed at the life that you see, even with a plastic lens of a toy microscope. Time went on, and I tried to excite my parents in this hobby of mine to study creatures crawling across the glass slide. But my father was rather skeptical that I could see anything with this toy. I remember being very offended. I decided at that moment to save my earnings. I used to babysit, mow lawns, and deliver newspapers. I wanted to save the princely sum of $100, which in 1962 was a lot of money, to buy a real student professional microscope. But I could never quite get the $100 that I needed because my mother kept borrowing the money and I couldn't get my microscope. So one Saturday morning when I finished mowing a neighbor's lawn, I rode my bicycle to the local police station and I told the duty officer that I was running away from home because my mother was stealing my money and I couldn't get my microscope. They called my father into the station. He talked to the captain and he came out. He was obviously very angry with me. But that afternoon, we went to a local pawn shop in Long Beach, and I got this, a Bosch & Lomb student microscope, which became my pride and joy through the rest of my high school years. At UCLA, they had better microscopes, so I left that microscope behind. And I became interested in chromosome replication, which I took to my graduate career with one of the great biochemists of the 20th century, Arthur Kornberg. And from him, I learned how to take apart a very complicated biological problem piece by piece in a systematic biochemical analysis. I decided to change fields to move into an area that was much less well developed, where I thought I could make my own name with discoveries. And so I chose to study a process that had only recently been understood at a very descriptive level, and that is how protein molecules, which are all manufactured inside of cells, how some substantial fraction of them get exported outside of the cell. So we make about 100,000 different protein molecules that do all of the chemistry and structure of life. But about a quarter of them are packaged and exported outside of the cell. You know these molecules. They are hormones, insulin, antibodies. Everything in our blood is secreted. Liver produces lipoproteins that are secreted. How do these get out of a cell? Well, a machinery has evolved to do this, which I will describe. First, here is a picture of the 23 pairs of human chromosomes. Every protein is encoded by a gene. Each gene actually encodes several different proteins, and they are all made inside the cell, and some have to escape. The membrane that surrounds a cell is a rather interesting structure. It's a two-dimensional fluid of lipid molecules. The red structures that you see here are called phospholipids. They have a polar head group that is exposed to the outside of the cell and another polar head group that faces the inside of the cell. But the interior of a membrane consists of apolar, hydrophobic fatty acids that like to hang out together, and they create an insulating barrier around the cell. But at room temperature or at body temperature, these lipids are mobile, and thus a membrane that surrounds the cell is actually a two-dimensional fluid in which the protein molecules that are embedded in the membrane that do all the important functions of membranes can bob and weave, they can spin about, they can move laterally around the plane of the membrane. So the challenge is to understand how a structure like that, not the usual covalent structures like proteins and nucleic acids, can be put together. Well, what we know about these membranes in cells came until the 1970s, largely from studies using microscopes, particularly a high-powered microscope called the electron microscope. And with that, you can cut a thin slice through a fixed cell, and you see an example of such a cell here. This is the beta cell of the pancreas. It organizes the production and capture and secretion of insulin molecules. So this cell is loaded with granules. These are all membrane enclosed granules in which crystalline forms of insulin collect and wait until you have a meal. When you have a meal, you begin digesting carbohydrates, and that elicits the secretion of insulin. These granules migrate to the perimeter of the cell and deposit their content outside of the cell. Insulin courses through your blood and allows the glucose molecules that you produce during digestion to be taken up and stored for future use. So these cells are just one of many examples in cells that are organized for this process of secretion. Here is, for example, the beta cell at higher magnification, where insulin granules move to the perimeter of the cell just when you start to have your meal, and the membranes merge. The membrane surrounding the granule merges with the plasma membrane in a process called membrane fusion. And as a result of that, insulin is discharged outside the cell where this crystal dissolves and the insulin courses through your blood. We'd like to know how this process works. It works everywhere in our body. Here's another example. This is a nerve cell touching or close to a muscle cell. These cells interact mediated by chemical neurotransmitters that are stored inside of these little granules. They're called vesicles, in this case synaptic vesicles, that carry neurotransmitters like dopamine or serotonin or acetylcholine. And they communicate, in this case with the muscle cell, by this process of membrane fusion, where the vesicle touches the membrane, merges, and then its inside is discharged to the outside of the cell, where it then interacts on an opposing surface to stimulate muscle contraction. This happens in the brain. Everything about us is controlled by this controlled release of chemical neurotransmitters. There are over 10 billion nerve cells in the brain, which make in excess of 1 trillion different connections among themselves. Each of those interactions informs a process of synaptic transmission and information flow that is basically everything about us, our memory, our thought processes, our intelligence, our behavior, our athleticism. Everything is governed by that careful flow of vesicles. Now, when I began my interest in this, when I started my career in Berkeley in 1976, it was known that this pathway existed in all cells that have a nucleus, in all eukaryotic cells, but nothing was known about how it actually works, how the process is organized. What are the machines that allow these membranes to be built, vesicles to be produced, and how these membranes fuse? Not a single protein or lipid molecule had been ascribed a role in this process. And I think it was largely because the tools that the cell biologists used to study this process involved, at the time, organisms that were not amenable to the tools of molecular biology. We had animal cells or animals where one could inspect cells, one could break cells open, but the tools that were used to understand genes were not yet available in these systems. And so I decided, when I began my career, to turn my attention to a eukaryotic organism for which these tools had been developed, and that is a simple organism, baker's yeast, Saccharomyces cerevisiae. It's a model eukaryotic cell. It has all of the machinery that our cells use to carry out these processes inside the cell, all of the machinery to replicate chromosomes and to make RNA and for ribosomes to function. And it has, in addition, very powerful tools of genetics and now gene cloning that were not yet available for more complicated eukaryotic cells. It's known, for instance, that when you grow yeast in the laboratory in a rich culture medium, if the conditions are right, the cell decides to divide, and it does so by growing a bud on the surface of the mother cell. It adds new material to the bud surface to enlarge it in preparation for cell division. 90 minutes later, the daughter cell is the same size as the mother cell, and it sets in a division plane that separates the two cells. And the process can go on exponentially as long as the conditions of nutrition are correct. Now, looking inside of a yeast cell by cutting a thin slice and using the electron microscope is not as rewarding as studying a pancreatic beta cell. That's because it's so busy making proteins that it is very efficient in its process of export. What you see mainly when you look through a yeast cell are wall-to-wall ribosomes. These are busy making the 6,000 different proteins that a yeast cell uses to grow and divide. But there are organelles. It's not a bacterium. It has a membrane called the ER, endoplasmic reticulum. It has a structure equivalent to the mammalian lysosome. In other slices, you can see a nucleus with a membrane around it. You can see mitochondria. But my attention, and specifically the attention of my star new graduate student, Peter Novick, and I, focused on what others had seen, and we saw as well, a collection of small vesicles, very similar in appearance to those that are in the nerve terminal, that were known to carry not insulin or neurotransmitters, but the enzymes that a yeast cell uses to scavenge molecules from its environment. We guessed, as our opening gambit, that these vesicles not only carried enzymes out into the environment to scavenge things, but that the membrane of the vesicle itself could be the building block for the growth of a yeast cell. Thus, by a process of membrane fusion, every step would enlarge the plasma membrane bit by bit until it got to be the size of the mother cell. Now, there's a very valuable prediction that comes from that idea, and that is that if you cripple any of the genes that inform this process, if you create by random mutation something that is a defect in that gene, the cell will die. That is, this process would be essential for cell growth. Now, there are ways that microbiologists have used to deal with cells that die like that. If you have a dead cell, there's nothing you can do with it to study its growth. But there are ways to look for mutations introduced randomly when you expose a culture of yeast to a chemical that causes mutations, to look for a mutation that introduces a single change in the molecule that makes it thermally labile. Yeast cells will grow at a range of temperatures. But if you have a mutation that cripples a protein partially, sometimes the cell will be fine at room temperature, but the protein will lose its function, will partially or fully unfold when you simply warm the cells up to human body temperature. These are called temperature-sensitive lethal mutations. That implies that the gene that's been affected is an essential gene, but you can grow the cells if you simply grow them at room temperature. So I had the benefit not only of a good idea, but more evidence in favor of this membrane fusion process. Here's another view of a yeast cell as it's growing. This is the bud portion. This is a bulge in the cell where the bud is emerging. And this particular technique cuts right through the middle of the membrane and you make a metal shadow replica of the cell. And you can see these dimples on the surface of the bud, kind of like craters, that reflect a membrane fusion event caught in the act of fusion, just as its content is being discharged. On top of this benefit, I had a fantastic new graduate student, Peter Novick, who joined my lab. He was a remarkable young man who was extraordinarily gifted in the lab. He is a reflection of the times. We all had long hair back then. He's now virtually bald, as I am close to being myself, but he was a great scientist, and he's now a fairly well-established professor at the University of California, San Diego. So he and I embarked on an effort to look for such mutations that cripple the cell in a temperature-sensitive manner, but which block the process of protein secretion when the cells are warmed to a high temperature. Of course, this was done in the context of Berkeley in the 1970s. Berkeley, the birthplace of the free speech movement, also was a place that was quite antagonistic to the use of animals in experimentation. There was a protest when they learned that we were going to be killing yeast cells in the course of our work. And this is some of the atmosphere at the time. We were able to convince the Animal Experimentation Committee that yeast are not sentient beings, and we have since killed billions of them, trillions of them indeed, with not a scream from any test tube in the lab. Well, one day, after Peter had isolated his first such mutation, called SEC1, short for secretion number 1, he showed that the cells accumulate enzymes inside the cell that should have been exported outside the cell. But when the cells are returned to room temperature, those enzymes are secreted. So it's a reversible effect. We were then eager to see what the cell looked like on the inside. And I'll never forget the moment he called me down from my office on the top floor of Barker Hall to the basement of the building where our electron microscope was housed to show me the following image that really changed my life. So these are cells, such as you saw a moment ago, that under normal circumstances have a small number of vesicles in the bud, but which under these circumstances, just after a couple of hours, as they're dying, continue to make vesicles that now fill the entire cytoplasmic volume. Thousands of them accumulate because of a failure in the last step of secretion. The gene SEC1 encodes a protein that is required for the vesicle to dock and then fuse at the plasma membrane. If the cells are returned to the permissive temperature, these vesicles restore their function and can fuse, and the cell resumes growth. Now, this was a project that then continued for many years. We isolated hundreds more such mutations. We were able to define the entire pathway with nearly 100 genes, each gene of which encodes a protein essential for a different step in this pathway. It became possible to clone these genes, but when we first did, they didn't look like anything then in a genome database because the human genome had not yet been sequenced. But when that happened, we found that all of our genes were there imprinted on the human genome, and that is the power of evolution. When you solve a complicated process like this, the bits and pieces are reused over evolution and are preserved. So much so that I use this as an example of that conservation. What happens to a yeast cell growing on the surfaces of a grape? is not unlike what happens in the brain of someone like Charles Darwin on the right. The machinery that nerve cells use to produce, move, and discharge synaptic vesicles at the nerve terminal are fundamentally conserved. Indeed, the SEC1 protein in yeast is homologous to the same protein in a nerve cell that executes the last step in neurotransmitter secretion. As a result of this, the biotech industry, which was growing up in the Bay Area in the 1980s, decided to use yeast as a platform for the manufacture of large quantities of clinically important proteins. And I consulted with a local company that introduced the gene for human insulin into yeast. And since it's just another secreted protein, yeast cells took it on as one of their own. And now, many years later, a company called Novo Nordisk, which owns the process, produces 1/3 of the world's supply of human recombinant insulin by growing giant fermentation vats, such as you see here, to produce metric tons of protein that gets secreted into the growth medium and can be easily purified. The same company was able to make a vaccine against hepatitis B by introducing the gene for the hepatitis surface antigen into yeast. It's a membrane protein. And so for any of you who've been immunized against hepatitis B, you've received little vesicles that were manufactured in yeast that house that antigen but have no infectious nucleic acid. So there is a very useful synergistic fundamental connection between the basic discovery that we do here at Berkeley and the application of it in technology. There will always be entrepreneurial people who will see the value of what we do and will invest in that and to useful purpose. And so I was grateful to have the opportunity to simply do my basic science and then to help a company use that knowledge. I'm going to turn for the rest of my talk to a related but different topic, and that is to the nerve cell. This is an image of what you might have of a nerve cell. There's a cell body with the nucleus. On one side, there are tendrils that correspond to dendrites that interact with the nerve terminal and receive information by synaptic transmission. On the other end of the cell, there's a long axon that can extend along the spine as long as a meter, finally terminating in these nerve endings, which can interact either singly or multiply with different nerve cells. As I said, there are over 1 trillion different connections in the brain that inform all processes that are characteristic of our life. These cells are the subject of tremendous pressure in the brain, and as a result, we suffer disproportionately as we age with neurodegenerative diseases such as Alzheimer's disease and Parkinson's disease. These are diseases, in the case of Parkinson's, rising at a dramatic and frightening rate. Parkinson's is rising in incidence even more rapidly than Alzheimer's for reasons that are partly genetic, but I think largely environmental. It's estimated that in a few years, over half of the new cases of Parkinson's will arise in China, not because of better diagnosis or because of genetic factors, but likely because of environmental toxins, some of which could be controlled, but which the chemical industry fights against their control. So we need, as scientists, to find a way to mitigate the damage of these chemicals. Well, let me tell you a little bit about what we know about Parkinson's, or what we've learned in the 200 years since it was first recognized by a Scottish clinician. One of the major features of the Parkinson brain is the appearance in the midbrain or disappearance in the midbrain of a cluster of cells that produce the chemical neurotransmitter dopamine. These are called dopaminergic neurons. They can be seen in a biopsy of a patient who died for other reasons in the midbrain because these cells, in secreting dopamine, produce an oxidized form of the molecule called melanin. Melanin is the pigment in our skin, but can be seen even in unstained sections in the midbrain of a normal patient. However, patients who die of Parkinson's have largely lost these dopaminergic neurons. It's estimated that when someone presents with the symptoms of this disease, over half of these dopaminergic neurons have died. So we desperately need to understand why these cells are so vulnerable in a disease that otherwise is quite variable. Another clinician, a German clinician by the name of Frederick Louis, discovered on sections of the midbrain of patients with Parkinson's disease that some of the dopaminergic neurons have big garbage heaps of proteins and particles that seem to aggregate in a big portion of the cytoplasm of the cell. And they may, he thought, be the basis of pathology of the disease. These unfortunately cannot be seen in living patients until now. It's been necessary to only see them in biopsies. However, recent developments in PET scans suggest that we'll soon have a diagnostic probe that can be used to follow the formation and progression of these in diseased patients. Now, the protein that is the basis of this clump is a protein called alpha-synuclein, a brain-specific protein for which mutations predispose familial forms of the disease. Patients who have mutations in the beginning portion of this gene, there are 3 particular mutations that cause the protein to clump somewhat more regularly than normal. There's another rare form of the disease where the gene is copied 3 times. It's only normally present in 1 copy, but some patients have acquired 2 additional copies of the gene, in which case there's more of the protein that's made in the brain, and as a result, the clumps form more readily. So we want to know what it is about the formation of these aggregates and their accumulation to form Lewy bodies that may be the basis of the disease. A subsequent German clinician by the name of Brock investigated in a large number of patient samples of biopsies of patients who died of Parkinson's at different stages in the progression. Early on, he saw Lewy bodies, they're now called, in the brain stem, but also curiously in the olfactory bulb. Now, it's perhaps a coincidence, but there may be some causal relationship. But patients who eventually present with Parkinson's often reflect that deep in their past, they had an acute loss of sense of smell. And we think that it may be the invasion of these Lewy bodies into this region that may be a precursor of the disease. But as the disease advances to the point of movement disorder, these Lewy bodies populate other regions of the brain, particularly the region of the substantia nigra in the midbrain, but also migrate into the cortical regions where sleep and motor disturbances are more prevalent. And then finally, in late stages, these are all over the brain, doing damage not just in the substantia nigra. And it is unfortunate that a large fraction of these patients develop dementia, a different form of dementia, not similar, but still as debilitating as that's seen in Alzheimer's disease. Well, there's another organelle that is implicated in Parkinson's disease, and that is a very important organelle in most of our cells called the mitochondrion. These are images. This is a cross-section of a mitochondria. Mitochondria evolved from bacteria. The ancient progenitor eukaryotic cell happened to swallow up a bacterium, and they began to cohabit in a symbiotic relationship, and now almost all the cells in our body have an organelle of bacterial origin. This bacterium is a powerhouse of the cell. It produces most of the ATP, the energy currency that drives the chemistry of life. And it's a carefully guarded organelle. The cell goes out of its way to protect this organelle and to get rid of it if it's damaged, because if the mitochondrion is damaged for whatever reason, its little chromosome, which it houses, escapes into the cytoplasm, and the cell sees that as foreign DNA, and that turns on a cascade that ultimately leads to cell death. In addition, within these infolding membranes of the mitochondrion, there's a last step in a process called electron transfer and oxidative phosphorylation, the last step in the manufacture of ATP, where a protein complex in this membrane holds on tightly to an oxygen-free radical that is part of a reductive force that makes water as a final product of respiration. That free radical, if it happens to escape from a damaged mitochondria, wreaks havoc in the cell. It can diffuse over long distances. It breaks chromosomes. The cell has to get rid of a cell itself if the mitochondrion is not handled. Now, of course, over evolution from 2 billion years ago, cells have evolved a way of surveilling mitochondria to find if they're damaged. They do so by putting a special sort of eat me tag onto damaged mitochondria, which are then recognized and enveloped by a membrane that delivers the damaged mitochondria and other bits of cytoplasm to this organelle called the lysosome, which is kind of the digestive tract of the cell, where the damaged mitochondria is obliterated into its amino acids and sugars and nucleotides, and thus any damage is mitigated. So this process is essential, particularly in the nerve cells of the substantia nigra, for reasons that will become apparent in a moment. Interestingly, there are two genes in the human genome that code for enzymes that are responsible for surveilling and putting this eat me tag on the mitochondrion. And mutations in either one of these genes produce a familial form of Parkinson's disease, which means that these genes, which operate throughout our body, are particularly vulnerable points in the viability of a dopaminergic neuron. Now, why is that? Why are these cells more sensitive, more vulnerable than other cells in the body? Well, an image of the cell may give you an impression of why this is so. In contrast to the image I showed you at the outset of a cartoon of a nerve cell, this is a dopaminergic neuron. It has a cell body, it has dendrites, it has a single axon, which terminate in nerve endings numbering in greater than 1 million. That means this one nerve cell can make 1 million different contacts with cells in another adjacent region of the brain that controls movement. This cell is always on. It's always busy. It requires a constant flow of ATP. Any damage to the cell that cannot be sustained propels it to death. That's why we think this cell is the focal point of these diverse processes that affect its viability. Now, I've told you this story. It bears only a minor connection to the research that I've done in my lab over so many years. So why would I turn my attention to this? Well, unfortunately, I have a personal story to share with you, and that is my wife, whom I married when I was a graduate student and with whom I raised a family in El Cerrito nearby. We were a happy family, perfectly healthy, 2 healthy children, grown and gone. But at the tender age of 48, she presented with symptoms of Parkinson's disease. In her case, it wasn't the traditional tremor that you associate with this illness. She had some aches and pains, but there was a very curious behavioral problem that she noted. One day she showed me in the evening, she said, something's very wrong. She wrote her name on a piece of paper, and it was tiny, tiny print. It was so small that I couldn't have reproduced it. I said, well, just make your letters bigger. She says, I can't. I said, well, that's weird. So she went to a neurologist and asked what could be the cause of this. And he said, well, it could be Parkinson's. It's a loss of fine motor control, not some gain of function. Well, how will I know if that's what it is? Well, you take a pill, basically a replacement for dopamine, and we see how it helps or not. And I'll never forget the call I got one day in my office. 2 hours after having taken a pill, all of her symptoms were gone, melted away in just a very brief time. That was good, but it was unfortunately, of course, bad news. It meant that she definitely had Parkinson's. Initially, in the early years of the illness, it could be well contained. Most people didn't know she had Parkinson's. This medication helped her. Parkinson patients, as they evolve with the disease, often it's the medication as well as the disease that alters their movement. You've seen people, they have these kind of awkward, jerky motions. She didn't mind this. It made other people feel uncomfortable, but it was a form of exercise for her. But of course, there were other things that accompanied the progression of the disease, very serious leg cramps and a cycle of on and off that could not be controlled by medication alone. She then had a neurosurgical procedure called deep brain stimulation, where electrodes are implanted bilaterally to control a region of the brain called the subthalamic nucleus, which in turn regulates the awkward movements associated with the disease. This worked really well for her. Her awkward movements were erased. Her cycles of on and off were leveled. The aches and pains disappeared. But within a year, it became clear that the disease had advanced because she developed signs of dementia. This dementia then progressed inexorably over the next 6 years. There were, nonetheless, life went on. We had happy moments. Here she is sitting with my father on the day of my Nobel Prize at the banquet in Stockholm. She was pretty much with it, but she needed constant care. I had my whole family there to care for her. But as we got on the plane to leave Stockholm, after a week of these festivities, she went to the bathroom and came back and said, I was waiting in line and I was talking to some fellow, and he asked me what I was doing in Stockholm. And she looked at me and she said, I didn't remember. What was the name of what we were doing? That's how bad it was. Well, it got worse. I had access to neurologists who understood her particular form of the disease. I would always ask what I could do to prepare myself. I knew from the outset that there would be nothing to do to mitigate the disease, to change the arc of the disease, but what could I do to prepare for what was going to come next? They always had an answer, and they were always wrong. It became impossible to communicate with her. She would forget what she was going to say, so she was silent most of the time. She would sleep all day if I allowed it. She was catatonic to the point where it was difficult to give her medications. She became incontinent. She needed full-time care. And then, because of pathologically low blood pressure, which accompanies the disease, she died in the middle of the night in 2017. Her physician said it was likely heart failure for lack of oxygen perfusion to her heart muscle. I should have said, well, thanks for letting me know, but there was no way of knowing. No one told me that this was a possible outcome of the disease. That's the frustration, both for the patient and especially for the caregiver. There is nothing that you can do to prepare. The disease progresses at different rates. It changes one's behavior in unpredictable ways. It's a source not only of anguish, but frustration. Well, out of this grief came something quite surprising. When Nancy died, I was approached by the chief financial officer of the Sergey Brin Family Foundation. Mr. Brin is co-founder of Google, enormously wealthy, but unfortunately, he has a family genetic history of Parkinson's. He and his mother share a mutation, a dominant mutation, in a gene called LRRK2, LRRK2, one of the 20 different genes in familial forms of the disease. This mutation is quite penetrant. His mother is quite ill, advanced illness with Parkinson's, but Brin himself is asymptomatic so far, but he's at risk. And as a result of his risk, he has over many years invested hundreds of millions of dollars in an organization called the Michael J. Fox Foundation, a major philanthropic arm that supports patient groups and clinical research on Parkinson's, but which had a very small budget for basic science. He decided, through his own knowledge, that what we really lacked was a fundamental basic understanding of the means by which the disease arises and progresses. Why is it that these dopaminergic neurons die? What can be done to arrest the progressive death of these cells? What is known at a molecular and cellular level? Not very much. So I was asked shortly thereafter to begin to lead an effort to understand and develop a program on the basic science of Parkinson's. So let me tell you a little bit about that organization. It's called Aligning Science Across Parkinson's. We have the generous benefit of Mr. Brin, who's invested over $1 billion now. We've developed teams of investigators to try to put the puzzle together. This is the puzzle as we might have seen it at the outset. There are a few pieces that are known to be bad that participate in the disease, but the disease is much more complex than this. There are probably 1,000, this is probably a puzzle that has 1,000 pieces with nothing on the face of the piece. Putting that together is really tough. So we decided that the best way forward was not to invest in individual principal investigators, but to find teams of scientists who already work together, who knew what it means to have a give and take relationship, to not always insist on where there would name on a list of authors in a publication or where the paper would be published. Teams who really were integrated. And so we've had, after now 7 years, the first group of funded teams. We had 35 teams with 163 laboratories in 80 different institutions in 14 countries, well-balanced teams with young investigators who had not worked on Parkinson's disease or more experienced male and female investigators. We've just launched the second and last 5-year program with another 31 teams that are just being given notices of award. It's been an incredibly rewarding experience to build these teams, to see people meet each other who had never interacted before. Most of our work is conducted online, but we had our first in-person meeting in the Bahamas, and these are all the PIs, the principal investigators of our initial team. And here I am in the front row next to my partner in this effort, Kemeny Riley, who's been the executive director of our program, and then the senior staff from the Fox Foundation, with whom we work hand in hand in this journey. We have 5 more years to go. We already have a number of leads. Our goal is to identify novel targets that can be applied by the pharmaceutical industry to develop drugs that really get to the core of the disease. And when I walk away from this effort, I'm really very confident that we will have the beginnings of a treatment that will bring this disease to an end. Thank you for your attention.
Thank you so much, Professor Schekman, and thank you for such an interesting talk about the machinery in cells. It's really like we don't think about how it could be so analytical about in that way, and how it has tremendous impact on human health and how we can treat diseases. So I'm going to open it up to questions from the audience, from students here in the room. If you have a question, please raise your hand and we will give you a microphone.
Not all at once, though.
Still processing. There's one question in the back that would be.
Hello. Okay, very nice. First of all, this was definitely one of the best lectures I've ever attended, so really amazing. And my question would be, what do you think, how long it's going to take for the research and the pharmaceutical companies to figure out a way to finally stop Parkinson's disease?
I'll give you a couple of examples of progress that have been made in recent years and are continuing. This gene that I told you that is mutated in Mr. Brin and his mother is an enzyme. And interestingly, the mutations that cause the disease are dominant. That means that they are gain of function mutations. And the enzyme happens to be an enzyme that takes a phosphate from ATP and attaches it to another protein. So it's called a protein kinase. So the goal of the pharmaceutical industry has been to find drugs that would dampen that activity down back to normal level. There are over 30 different biotech companies that are in process. One of them is in San Francisco. It's a company called Denali. They have a drug that is brain penetrant that allows them to dial down the activity of that enzyme. And they are now in phase 2 trials, and we're looking for a readout of the phase 2 results in the summer. So fingers crossed. Now, of course, this is one gene. It's only a small fraction of the Parkinson patients. We'll be happy if it works on that number of patients. But there's reason to think that enzyme may have a role in even the sporadic non-genetic forms of the disease. Another quite independent strategy is to find a way to replace the dopaminergic neurons with freshly minted neurons that can be made in the laboratory. So it's possible to take a kind of a stem cell and turn it into a dopaminergic neuron in the laboratory. And there are 3 companies, one of which is a company called Blue Rock in New York, that are in clinical trials where they've created these cells in the laboratory and they've implanted into the substantia nigra under conditions where in experimental animals, the processes of these complicated cells reach out and make connections to adjoining regions of the brain, at least in animals. So far we know that these implants have been tolerated, and there's some indication that they may be effective. This particular company has been given permission to go right from phase 1 to phase 3, quite unusual. So again, fingers crossed, we'll see. And there are efforts like this that are informed by the research that are very different than what the pharmaceutical industry has done till now, which I'm quite confident in by the time, as I said, I walk away from this, these will be in the clinic.
Thanks.
Wonderful. Any other questions?
There's a
One on the
question
left
over there.
here.
There's a question here too.
Right.
Thank you, Professor. Later on, this class is going to be presented with the work of Jennifer. Maybe you could explain to a non-biologist how her work on gene editing.
Yeah.
How does that
Yeah.
affect your work?
Okay, so of course she's a colleague of mine. I've known her well. She has a big operation. She runs an institute, the IGI, here on campus. One of her colleagues is using a version of the CRISPR technology she invented to turn down the level of production of this protein alpha-synuclein. So we know that if you kick that gene out, it's probably not good. But if you can just turn it down, just like I said with this enzyme LARC2, if you just turn it down a bit, maybe twofold, the lower level of expression will diminish, reduce the rate of accumulation of these clumps of that protein. So that technology absolutely has an application here. The challenge is to get the gene editing tools into the brain, specifically into the region of the brain that's affected here. That's a really big challenge. It's easier if you're just taking a pill to have it diffuse into the brain, but to deliver gene editing tools to the particular region that would have the salient effect is a big challenge. But of course, she has members of her team who are working on that as well. So she's terrific. You'll hear more from her.
Thank you. We have a question in the back here.
Thank you for the lecture. You had one slide where I think the statistic was that in 2030, 57% of Parkinson's disease will occur in China.
Yeah.
Why is it so much more than in India? I think it was only 8%.
Yeah. That's a question I just asked because I was just in India and the pollution there is unbearable. I think it probably is just as prevalent there, but it may not be being reported as well as it is in China. But sure enough, if you ever have the trauma of visiting New Delhi, I mean, it's unbelievable at many levels, but the air is I grew up in Los Angeles when it was pretty bad. There's nothing like what you see in New Delhi. This has to have health consequences there. I wouldn't even want to visit the place.
Thank you.
Great. I think we have time for one or two more, depending on the length of the question.
Hi, Professor. What do you think was so effective about your approach in bringing together teams with existing chemistry versus bringing in the top researchers and other?
Yeah. Well, we looked for people who had already been collaborating, either on Parkinson's or something related. There are grants that are issued by government or private agencies that ask to build a team. But, when you dangle money, teams suddenly appear. These are among people who haven't necessarily worked together. So they don't necessarily know. When you have a meaningful, enduring collaboration, it's because not only is the science important, but the personalities blend. That's really, really important. And so we look for teams that had already built that, who could prove that they had published papers together, that they'd written grants together, that they'd organized meetings together. And our way of, as we monitor these teams, when they joined our network, we developed tools to follow their behavior to see whether they work well together within the team, but among teams too. And so before we even decided whether what they published was valuable, we issued report cards to these teams about their team behavior. And some of them were shocked when they were found wanting. So now going into the next round, we really think we know what makes a successful team. That, I think, is the key.
Great. Thank you so much. I think that's all. Sorry. I think that's all the time we have.
I'll be around. You can ask questions.
But Professor Schekman will stick around after this One more round of applause, please.